Dr. Heiniger here on the screen and ready to go from North Carolina. We met him through a consultant named Denny Wildman from Farmdale, Ohio, many years ago now. Denny has worked with Dr. Heiniger with a lot of plots. Dr. Heiniger does tremendous work in North Carolina and beyond. They run many research farms. I make this state here, but I think at least 7 or 8 different research farms across the state. They look at all kinds of different practices and crops and genetics. And we've been fortunate enough that he's had enough interest in our products that he's looked at our biological along with many other biologicals on multiple sites, replicated plots for many years. So we're very thankful. He's been very gracious to help us. He's highly respected amongst the corn industry down there in North Carolina and beyond. He's as dedicated as anyone we've ever met. He cares about the farmers in the region, test what they want him to test, research it, look at it, get the data, get the proof, and present it to them. He does more meetings than anyone I think I've ever met, sharing his information across the Carolinas down there. So with that, we'll turn it over to Doc Heiniger.
Well, thank you. Can you hear me okay? Are you hearing me fine?
Yes.
Good. All righty. Yes, well, thank you for that kind introduction. I'm sorry I couldn't be there actually in person. That's my preferred mode of doing meetings. But, you know, these days, why the opportunity to cross time and space is sort of enticing so that we can share information a little bit better across the regions. Indeed, I'm the corn cropping specialist in North Carolina. Actually, my title is cropping systems specialist. I've worked in wheat, cotton, small soybeans, and some other crops while here at NC State. And indeed, we have a whole range of research stations across the state, like many land-grant universities used to have anyway. And we utilize that. Our state is quite variable as far as soil types and environments. So here I'm showing a screenshot of the flatlands of eastern North Carolina. These are muck soils, so very dark organic soils in what used to be poorly drained regions of the state. And we have plenty of sands, as you probably well know, coastal plain, sandy loam soils. And then we get into clay loam soils in the Piedmont. And of course, in some of the mountain valleys in the western part of the state, we have some very productive alluvial soils out there. So a wide range of environments. Typically, our northern latitude is similar to the southern. Well, I came from Kansas, so I'm used to the regions of Kansas. So our northern latitude is similar to southern Kansas. And our southernmost points are sort of like North Texas. So indeed, sort of that region of the country, we get a lot of rainfall, 40 to 50 inches. So anyway, enough about me and some of these environments I'm going to show you. Let's talk a little bit about what I'm excited about. I think the future of agriculture should be excited about. And that's understanding and incorporation of biology. I'll call them biostimulants here, but biology in various forms in crop production systems. This is some of the work we've been doing. This picture here actually shows some of the concerns I have. You can see 8 rows of corn here that looked a little yellow versus corn beside it. It looks very nice in this situation. This was planted late March in northern North Carolina. And why? Yeah, we've got plenty of nitrogen, phosphorus, potassium in these soils. There's no reason we shouldn't have this kind of differential in crop look. So indeed, I think biology offers us some things that we need to have here. All right, let's see. There we go. All right, so what am I talking about when I use the term biostimulants? Well, there's lots of different things that might fall into this category. Humic and fulvic acids could be categorized as a biostimulant. We certainly have looked at some of those in cropping systems here. Biological mixes of different types of biology, everything from fungi, bacteria, algae. That's sort of where SP1 has fallen into this lineup. Live or stabilized bacteria, fungi. Certainly SP1 has some of that as well. Enzymes are biological molecules. There's a whole range of those kind of products that are touted on the market. And then certain things like growth regulators are actually a biostimulant when you come to think about it. So why I just mentioned that I thought these were the future and why I'm excited about that. Let me just tell you a story of why I'm excited about this. Yeah, this actually comes from 2017, 2018. We were very interested in following behind people like David Hula and Randy Dowdy, who had got 4, 5, 600 bushel to the acre corn yields, understanding what it took to make high yield corn in terms of limiting factors. Was it light? Was it nutrients? Was it water? So indeed, we started a series of tests in conjunction with other scientists at NC State to try to understand that. And I'm just showing you here on this screen some soil test results from 3 fields at the Kinston Research Station near Kinston, North Carolina. This is a sandy loam soil. You can sort of tell that by looking at CECs there and weight to volumes. This is a sandy loam soil. The point I'm making by showing you this information is you could take a look at nutrient content there, particularly phosphorus here. That we use index. I don't know that anybody else in the whole country uses index. We do that because of these high organic soils here. But you just take an index times 2, and basically you come up with how many pounds to the acre that element you've got for phosphorus times 3 for potassium and so forth and so on. I show some of these conversions here. That's 500 pounds per acre of phosphorus in these fields here. This is not unusual in North Carolina. We get a lot of animal waste here, a lot of pig and poultry operations. So we have manure. So these soils tend to have fairly good nutrient content, and that's certainly the case in this situation. If we had any weakness here, it'd be our pH is a little bit on the weak side in this situation. So we have a good fertility situation here in this field. We supplemented this field. We put on ammonium nitrate, or DAP, diammonium phosphate, sorry, at about 80 units of P2O5. We put on some micronutrients. We used a starter fertilizer. This field got about 350 pounds to the acre of nitrogen, so plenty of supplemental nutrients here. And here's the yields. Now, they're not bad. You can see that water should not be limiting here because we have no irrigation of half a volume, then full irrigation, and then we had a fancy sensor system out there monitoring when that crop needed water by monitoring soil moisture every half hour or 15 minutes, I think, actually, on the day. Those are good yields, 217, 204. That's okay, but that's certainly not what we were interested in. We were interested in something over the north of 300 bushels. So what we went in and dug in, and what was our limiting factor here? Well, believe it or not, it was nutrients. It was nutrients. Take a look. These are tissue samples. A graph here, a 4-bar graph, where we show tissue samples taken at B6 in corn, silking, dent, and maturity. And you can see the upper left one there is leaf nitrogen. That little gray boxes represent sufficiency levels. And you can see by dent stage maturity, we're well below sufficiency level. Actually, on average over our samples, we were at the low end at silking. Probably more shocking than nitrogen is phosphorus. We aren't at sufficiency level at any stage of the cropping system, and you can see other nutrients there. I could put up the whole range of nutrient graphs that we have here. The question that became obvious is why? How in the world can we have all that fertility in the soil? We added fertility. We tried to do a good job of balancing nutrients, and yet not meet the crop requirement for nutrients, particularly phosphorus, in that plant. That just stuns us and leads us to the conclusion that when we talk about nutrient environment for crops, really, we need to have 5 R's, not just 4. We got the right rate, time, source, and placement to apply fertilizers or nutrients, I think we really have missed the fifth R, which is the right environment for uptake. In corn, that environment is those first 5 nodal roots, which form the massive um, root reservoir or root mass that's required to explore the soil and find nutrients. This little picture I've got here with this is another good example of what I'm talking about. Here's a cornfield on, again, a sandy loam soil north of Goldsboro, North Carolina, planted April the 8th. We put 50 units of nitrogen here at planting time, and yet we've got yellow corn. And you could certainly see we got some plots where we did something there. You know, how do we have yellow corn here? Obviously it's roots. Obviously, some of this corn is not exploring this soil, whilst other treatments have allowed that corn to explore that soil. So, how does this all relate to biostimulants and soil biology? Well, I think what our conclusions from all of this have come to is we need to be a better steward of the biology of the soil. We need to understand and utilize instruments, if we can, to improve our soil biology. Importance of soil biology for soil health. This I just obviously got off a photo credit off the internet here, but you can see there the aggregation of the soil, the utilization of fungi by those root systems to help further explore soil particles and pull nutrients into that root mass, the water flows environments of the root system all have to be functioning in order for us to get these nutrients that in many cases are in the soil. I mean, I get this from farmers all the time. I apply all this fertilizer. I'm trying to increase my yield. I seem to hit a limit at which I can no longer get yield increases. What's going on? Why are they not? Why is my field's not responding to what I'm doing? Well, it's not responding because we forgot the biology component of our soils here. And I think most of us probably realize this intuitively, and that is our current state of soil biology is not very good. We've tilled over time, reduced carbon and organic matter in these soils, reduced the environment for these bacteria, endomycorrhiza, protozoa, algae, reduce those food sources for those organisms, which further then causes loss of soil structure, which leads to then water saturated, poor air infiltration, anaerobic soils, which then in turn further reduce bacteria, endomycorrhiza, and other biological organisms in our soil. It's a vicious cycle that we are entered into here with our current thinking on how to manage soils. Even under tillage system, low tillage or no-till systems where we put in or returning large amounts of biomass, we often see soil become more compacted, not less. And that again is because we're not returning this carbon to the soil. We no longer have the environment to actually utilize the carbon in those soils. So indeed, oftentimes we see, even when farmers move toward less tillage or heavy residue systems, why they don't see much benefit in those systems. So I'm going to talk a little bit about some agri-energy products here today. And while I do so, I want you to keep in mind some of the things that I think about when I try to measure efficacy of these products. You know, the direct way would be just to go in and measure biological activity. In other words, see if those organisms that were applied to the soil are actually replicating and increasing their presence in our root systems. Of course, we can look at plant biochemical responses. These are certainly possible, but very difficult because biology, like most environmental organisms, vary based on temperature, moisture. So indeed, taking a spot sample in time is a very tricky situation, depending on what time you would take the sample. Is it dry, hot, or is it wet and cool? That all those factors come into play to try to actually monitor or measure. Of course, the samples themselves are extremely expensive. So the next best way to look at efficacy, and one of the ways that I try to do so, is measure impact. Are we getting increases in nutrient uptake? Are we seeing an improvement in plant physical response? And we'll see some of those measures here in this talk today. And what we oftentimes settle for, of course, is yield response, which is an aggregation of all the impact of the environment through the season. So, indeed, keep that in mind here as we look at some of the results. So, let's talk about nutrient uptake. As I mentioned earlier, this is where my interest in these products is coming from. Can I improve the root soil interface and improve nutrient uptake? Well, let's just compare with what our traditional approach is to try to improve nutrient uptake. Here are some of the things that I think have some impact: drainage, starter fertilizer, nitrogen, micronutrients, and then biological activity. So let's look at our traditional approach to this would be to put fertilizer, particularly phosphorus fertilizer. So let's see how that works here in this situation. Again, a Norfolk sandy loam, coastal plain, North Carolina. We put these treatments out. This plot was planted about the 12th of April. Yeah, in 2020 here is a couple years ago. Here's the check, no starter. The 4 rows behind that sign are 50 feet long where no starter has been applied. You can see the color of the crop. You can see I pulled up. Now, again, this is not a field lacking in nutrients. The phosphorus index here, again, or pounds per acre, is up above 250. Nutrients all across the board. Even the pH here is 6.1. So almost ideal soil test for making good corn at this location. You see, the root system doesn't look too bad. A reasonable crop if you didn't know anything better. Here's what happens if we put a little in furrow, 624-6 blended fertilizer, applied at 3 gallons in furrow. Looks a little better. Obviously, there's some nitrogen being picked up there, either from the fertilizer or enhancing root systems. You can see a plant on that board. I put a plant there on the board to illustrate the starter or the no starter plant, and then this 624-6. So the plant's a little larger. There's no difference in the growth stage here. We're just adding more cells. So indeed, the leaves are wider or longer. The root systems tend to be more prolific. So we're obviously increasing the energy in the plant and increasing cell division here when we do this. Here's probably the traditional approach to try to get, and you can see it certainly has had an impact here. 1027 or 1137.0 is oftentimes used. I'm using a 1027 because I could use some micronutrient pack with this to get some zinc. So you can see there a lot of growth, big difference in amount of leaf area, root biomass on the board compared to the no starter check there beside it. So again, obviously, these are methods for trying to improve root uptake. And there you can see what I usually look like when I don't look very good. So here's the impact of fertilizer on here's yield. And no surprise here, with those kind of differences in early growth turned into differences in yield. Now, this isn't always the case with starter fertilizer. We can actually get too much growth and then run out of water, and that can lead to negative consequences, actually, in comparison to the check. But in this case, we had some irrigation, so we weren't worried about running out of water by soaking time on these sandy soils, so we got a 50 bushel yield increase with those nutrients added. So a positive result. This is how traditionally we've handled these kind of issues with starter fertilizer. Well, how about biological activity? Can we turn what we had there, again, plenty of nutrients in that soil, we clearly are not getting them into the plant. Can we use biology to do this? So we're going to look at a couple things that AgriEnergy is associated. Here's the Myco-C MSTC treatment. You look at the active ingredients, it's bacteria in multiple forms, Linchenbacter, actin bacteria, fungi, the endomycorrhiza. So this is directly from some of their information that you can find about this product here. And again, the idea is to use biology to try to stimulate this root uptake. The other product we're going to see here is this AgriEnergy SP1. This has been a product that we've used extensively now for 5 years in testing. And it's, if you look at active ingredients here, it's got a range of bacteria. It's a blend of things, algae, bacteria, fungi. There are carbon substrates that support those, minerals and other types of things associated with the biology that typically can be gleaned in our soils. So these are the two products. You can see their active ingredients here. They're no fertilizer whatsoever. They're just biological organisms or molecules. And what's our goal here with using them? Well, it's just primarily to stimulate the root soil interface. Particularly, I'm relying on mycorrhiza, the avascular mycorrhiza in the soil, or with these products to form these interfaces with the soil and the plant root and promote these interaction, the exchange of protons with the root and allow available nutrients to be taken up by the plant. I'm sure you've seen many of these types of photographs. They're readily available. There's lots of information. Whenever I look at a product, I like to know how it works. I'm sure you do as well. What's the driving factors here? There's lots of information on the role of avascular mycorrhiza in promoting root function. Here's a good paper that was done by Tony Venn at Purdue and a student of his looking at improvements of the drought tolerance. So the DS in these graphs here are drought stressed, and WW is well watered. AM represents avascular mycorrhiza. So that's the black bar. The gray bars are the non-AM or vascular mycorrhiza systems here. And you can see leaf relative water content in the leaf is higher where we have the drought stressed, and the root protein concentration is higher. They had a number of graphs in this paper. We can find other papers that talk about the similar impact of vascular mycorrhiza improving water uptake and increasing root gram weight and protein. So, with all that as a background, to say what we're interested here in seeking, it's a product that can bring that kind of interaction to this field. So we put MST on, got 4 ounces to the put it on the seed at the time we planted it. And then we're using SP1. Now, this is not a high rate of SP1. It's only 1 gallon per acre. We're mixing with water and putting it in furrow at a 5 gallon per acre solution. Now, there's some interesting things to see here. First of all, I've got on the left, I've got the plant that on that board that was from the no starter check. So you can see these MST SP1 treatment plots are certainly larger. That's that plant on the right, the 3 that's on the board. The one in the middle is actually from the 1027-0. Now, it doesn't look as green, and I don't think there's quite the biomass in this 4-row plot as the 1027-0, but it certainly's got a lot more biomass than the no starter check. Here's what was really interesting as we looked at these plants closely. And that is, if you look at this plant closely, you can see this darkening around the roots. We're trapping soil here. There's a lot more mycelia and fungal growth associated with these roots as you look at that root compared to what you're seeing there from the no starter check on the right or the 1027-0. Those roots looked white when they came out of the soil. But these here, where we're taking these, digging these plants, we've got fungal growth on root surface. There's obviously mucilage or excretion from that root that's trapping soil against the root surface. And then there's you can see the fuzziness, the abundance of root hairs that are present in this root. So clearly some differences in how these roots are looking or functioning in their environment here. Here's what some of this corn where we tested MST and SP1. The other factor that we oftentimes see is at harvest time, we see less damage to these kernels from fungal ear rots. This is aflatoxin or fusarium ear rots. Here on the left, we have just a starter. That certainly those ears look nice. That cord gets off just like we just showed in a similar manner, but clearly the ability of that root to uptake things other than just nutrients, phytochemicals, enzymes, and produce different substances have some protection ability in the plant. And we've known this about biology for a long time, that there was benefits from plant pathology standpoint by having good soil biology. And certainly we see this as we compare these two sets of ears. So how did that perform there as far as yield? Well, every bit as good as the nutrients, the fertilizer that was applied here. So where did that yield come from? We didn't put any more nutrients here. Not like 10-20-7-0, where I put 20 more pounds of nitrogen and almost 54 pounds of P2O5. I didn't put any more of that there. So it's obviously coming from the soil. And in this sandy soil, we're clearly taking advantage of the nutrients that we already have locked into that soil profile. Here's another example of some data that we had from 2019, where we're looking at, I said earlier, that was a fairly low rate of SP1. Here's a 2019 example where we looked at some different rates, 1.5 gallons, 3 gallons, and 1.5 with a fulvic acid here. And again, you can see we had some nice increases in yield. So probably, if I had any criticism of that 2020 trial, is we may have been a benefit to increase the amount of SP1 we utilized in that trial. So what I'm trying to show you here, of course, is this idea of efficacy by looking at plant responses here. So we looked at plant response, we looked at the roots. Here's another way that I've tried to look at efficacy by looking at plant responses. So here we have a check, 10-20-7-0 with a Stoller product called Energy Power, 10-20-7-0 with Energy Power and SP1. You can see differences in plant size here. There's differences in these roots to some extent. You can see a lot more central roots in that SP1 plant, and there was a lot more root hairs in that root system. So similar, very similar looking roots. This is clearly not a sandy soil. This is a muck soil here. So this soil should have a lot of carbon and should promote biology in this environment. What we wanted to see here was actually whether it changed canopy temperature. In other words, another response to mycorrhiza improving leaf water content and therefore reducing canopy temperature. One of our primary problems here in the southeastern United States is we're high humidity. We get hot days during late June, early July. It's not unusual to have canopy temperature soar into the low to upper 90s or even above 100. Anytime canopy temperature in corn is over 95 degrees, we're basically going backwards. We're using more energy than we're producing with that crop. So you see it roll up, the leaves roll up, and we don't make much progress toward yield. So we're interested in trying to keep that temperature. Here you see that this SP1 added to this significantly reduced canopy temperature. Again, an indication of biological activity in the soil in promoting this water uptake in these plants. And then here is the yield. So a significant increase in yield. A lot of times when we see the ability to keep improved water uptake, we certainly see this response in yield in these crops. All right, so let me just show you what we've seen. So there's some examples looking at cases where we've tried to measure efficacy by measuring plant responses. Probably the most common way to look at products is to measure yield, and certainly in analyzing a number of products, which you can see here on this chart we have, why we look at yield over many sites. In this case, 2019, I have 3 different locations. Hyde County is a muck soil in eastern North Carolina. Sampson County is a Goldsboro sandy loam in the coastal plain, and Robeson County is southern coastal plain. Those are both sandy loam soils there. You can see we've tried a number of different ways to look at biology. The MST SP1, so now I'm using 3 gallons, a higher rate in these trials here. We're using a product from Nutrien called Levitate. It's a 5-15-5 with a biological catalyst called Accomplish. Some of you are familiar with it. Levitate with some Radiate, which is root. So all of these are biological stimulants. There's Accomplish alone, Albit, which is a plant enzyme stimulator. There's some Monty's liquid carbon products, a Midnight, which is a foliar application of nutrients applied at V6 typically. BioStrain was applied at planting. The liquid humic acid. There's that 1027O at 10 gallons, which is often my comparison of how good those products are performing. And then we have another Select Ag formula set of products. So many different types of things here compared to an untreated check. You can see the red here typically denotes significant difference from the untreated check. So you can see we don't always get a significant response in yield compared to the untreated check. You can see I have a numerical response here, but depending on how much variability in the trials, why we may not get a statistical response. But when we combine overall sites, the MST SP1 combination was the highest yield, significantly higher than the untreated check combined over a range of different conditions. This is one thing that I've liked about the SP1 product. A lot of these materials, like Accomplish, has got bacteria, but not an endomycorrhiza or a fungal aspect to it. The SP1 in particular, I think, brings something where, regardless whether your soil's low in or needs algae or needs bacteria or needs protozoa, you've got a product that covers a wide range of biology that fits that need across different environments. And that's certainly the case, what we see here. Here's another, that 2020, we did a similar studies trying to compare the impact of products here. So again, we've got 4 different sites here. Rather than use numbers, I use bars in this case. But I've got Beaufort County as a muck soil, Wayne, Duplin County are both sandy loam coastal plain sites, Surrey County is a clay loam in western North Carolina. The little hatch marks in this case indicate significant difference from the check. So Beaufort County, we're looking for the green bar here. Again, it becomes sort of confusing with all these products compared, but we're looking for the green bar. How often is the green bar significant compared to some of these other products? I can, it's like I just mentioned. For instance, a good illustration is heat shield. Heat shield tends to work best in certain types of environments. In other cases, the heat shield is a Hefty Brothers product. It's a strain Trichoderma endomycorrhiza, and it tends to work in some cases, but not across the board. You can see here the green bars are 3 out of the 4 locations in which I have a significant response from the MST SP1 product. In this case, that's actually better than the 1027O. Like I said, what I typically a grower would go to is let's put fertilizer here, and indeed, in this case, the biology or the biostimulant is improving yield more consistently than use of a starter fertilizer. Here's SP1 directly compared to the starter fertilizer in that year. Now, again, I'm using this Levitate with the Accomplish material in it. And you can see that we're looking at the SP1 broadcast either in furrow. We put 1 gallon in furrow. We put 2 gallons broadcast. We did a combination in furrow broadcast and then a 3 combination. And again, at the Beaufort County site or in Washington, both of these would be either silt loam or muck soils. Well, you could see we were getting more consistent responses with the SP1, or the SP1 when added to the fertilizer, which already had a biological component to it. So again, it shows the versatility of this product and how it performs across a range of environments, which again, as we talk about biology, that's been the key thing that I've discovered is that you've got to be careful in many of these materials what environment you're applying them to. I mean, the latest and greatest is these biological nitrogen fixation materials, the proven pivot bios, proven, and some of these other things. They work if they're in the right environment, but they will not work if they're not. And so finding a product that has the capability, a biological product, is very rare in the marketplace today. So here's MST, SP1, just to see if indeed the combination or one alone has beats each other. In other words, do I need both or can I use SP1 alone? And in this case, you could see that at this site in 2021, they both increased yield significantly when used alone. There was a slight increase when they were synergy when they used together. But clearly, in this situation, the bacteria that was present in both of these products was the key driver for the soil response at this location. I don't remember whether this was muck soil. I think it was. And then when is the best time to apply? Well, we've tried some different things. In furrow, V6 with the SP1 product. And what we've found is typically in fields with no prior application, an earlier application tends to do better. Now, I've got some data where we've applied SP1 previously that show a V6 application or a foliar application doing very well in comparison. But this data last year that we took here on a new field tended to say that the in-furrow application was effective. This was actually a SP1 product that we had on hand, so it wasn't fresh. So it shows the capability of the biology in these mixes to stay active. Finally, one of the things that interests me or what I've noted, and I was trying to get a handle on how to show this, is that where we've used SP1, we see a change in the soil. Now that's, I know, hard for many people to grasp onto, and it seems somewhat surprising even to us in the research, but that's what we're seeing. Here at the left, this is clearly a field that has had corn, and there's residue. This was corn from a previous year. We're coming back into the spring here. We're going to put some more plots on this site. But you can see on the left where we applied SP1, that soil is more granular. On the right, you see these chunks. Now you might think, oh, well, that's good. But no, that's compaction is what we had to jump on this shovel here to get this soil dug up here, having laid in the winter under heavy rainfall and conditions. Why this soil, you can see all these chunks and hard pan that's developed, whereas we're not seeing that in the SP1 soil. So clearly the biology has more than just a initial impact on this soil environment. So yeah, there's SP1. Now that's a total of 6 gallons. That was where we'd put the 3 different applications the year before, and there's no SP1 there. Here's what I came to. I went back and I looked at places like you just saw where we've used SP1 in the same plots or in the same location over time since 2018. and compared it to where we had not used SP1, no biological treatments whatsoever in those plots. Now, the black line is the check line there. So you're reading corn yield on the, this is a confusing graph. You're reading corn yield on the left and the amount of rainfall during the growing season, April through August, on the right here, because we know differences in yield are across years are typically driven by differences in rain or moisture, which is the most common outlook. If you looked at corn yield across time, it highly correlated to how much rainfall you're receiving during the season. So you can see that black line mirrors that seasonal red seasonal rainfall line here. That jumps up where we had that 20 some inches in 2021, which was the best corn year we've ever had in North Carolina here, and then right back down in 2022, which was one of the driest years in the last 10. But if you look at, and we've been getting, as you just saw, some responses at this site with SP1 applied. But if you look at the yield where the biology has been applied, you notice we're not getting the same correlated response with rainfall. It seems like we're basically increasing or improving the soil over time. And this is exactly, you can see our yields are now up in the 250, even in the dry year. I thought a year that we should have had does very common yields, we're still hanging in that 240, 250 range on those plots. So this is one of the things that I think we need to do a better job of quantifying is how these biological stimulants are changing our soil environment over time. We're reversing this degradation cycle that I mentioned earlier in this program and reverting back to a better situation. Let me wrap up. I've spent too much time here, I see now. Improving soil biology can improve root mass, increase soil tilth and health, and increase water and nutrient uptake and yield. We see impacts both from the plant physical responses as well as from the yield. So I'll take any questions if you have them. Happy to have had this time with you.
Oh yeah, we got time for a couple of quick questions.
Yeah. I'm curious about treated seed corn with our in-furrow biologically.
Yes.
And our trials. I gave up treated seed quite some time ago, but I'm really curious about that holistic approach. I'm really curious about the holistic approach to if we're going to get that rhizosphere biologically stimulated, making sure that like that treated seed out of it, or if there's a correlation there with just our pathogens, if they seem to be there. And then on that same approach, I've been using those same in-furrow biologicals as my fungicide and getting pretty significant results out of that. Is that part of that just building process? Is that why? I mean, are we balancing out those pathogens this way?
Yes, I think you're exactly right in that we're basically one of the things that I worry about using in-furrow, and we've tested in-furrow fungicides on corn, and one of the things that I noted in our testing is I don't like to put them in or close to the seed. I like them in a banded application or just using a furrow jet that pushes it off to the side of the seed. So I don't like them on the seed because what I think happens when you use an in-furrow fungicide on the seed is that initial root, you reduce the colonization of biology on those initial roots, and then you don't get that start. So basically, you're right in that sense. When you're using a fungicide, you want to balance it out with some a biology to quickly put the stimulate the good fungus, and while your fungicide is reducing the pythium, the phytophthora, and all these organisms that are common in anaerobic soils. That's where you find those organisms is when soil aeration is poor, and you don't have oxygen exchange. So yes, there's a balancing out. I agree with your focus of your question is right on track. we've got to be careful. You know, a lot of these materials, I worry about seed treatments, anything that is negative or can have a delusory effect on my soil biology. Even using some of these biological things like heat shield, without careful application, it cannot be mixed with certain things. If not careful, it's rendered ineffective because, of course, it's a pure strain of trichoderma. So you got to be very careful about mixing things. And I think that's true. This is why we're in the situation we are in. We went down this road. Oh, let's add a seed treatment and let's do that. Well, it got worse. So let's put more seed treatment on, so forth and so on. And that just reduced these beneficial biology in our soil. We've got to find a way to balance that, like we've just talked about, or at least reduce these. And I think we can once we have these soils back into a healthier, more aerobic state around that seed.
Okay, thank you, Doctor. One more quick question, anybody? Not, I'll pass it back to Dean, and then we're going to get our next speaker on the line.
Thank you, guys. Take care.
Yes, thank you, Dr.
Thank you,
Heiniger.
Dr.